Now let's look at see how we can maintain acid-base balance using chemical buffers. Now chemical buffers, or any buffer, is something that helps maintain the pH. That is, if I added acid to a solution that contained a buffer, the pH wouldn't change because the buffer, in a sense, neutralizes that acid. And so buffers help maintain pH. And we'll look at the different buffer systems, the proteins, the bicarbonates, and phosphates. They're all the chemical buffers that we find in our bodies. So first, let's get an overall look at the different buffers we have. There are both chemical buffers and physiological buffers. The chemical buffers are our first line of defense. That is, if a pH should drop in the blood, these, or in cells, or in extracellular fluids, these buffers kick in immediately and then help maintain a normal pH. So this is going to include things like the bicarbonates, phosphates, and proteins. But those buffers kind of get used up. We have limits on how well they work, and so we have to go to the second line of defense, and that's the physiological buffers. And that's how the respiratory and kidneys or renal mechanisms kick in and help in neutralizing or balancing our pH. So again, looking at chemical buffer systems, these again prevent rapid change in pH in the body fluid, and they kick in very fast. And they're different buffer systems for different areas. So we have intracellular fluid, that would be the fluid inside of cells, or the extracellular fluid, of course, fluid outside the cells. So inside the cells, we've got the phosphate buffer system that works really well. Extracellular fluid, we've got the carbonic acid bicarbonate buffer system that works very well. And then the protein buffer system is going to work in both conditions. That is, we've got three examples of where these are going to work. Hemoglobin is a good example of a buffer system that works really well in red blood cells, so inside or intracellular fluid. Amino acids can work both intracellularly and extracellularly. Wherever they're located, they can buffer. And then the big plasma proteins, of course, are extracellular proteins, so these are going to be effective buffers in the extracellular fluids. So we'll look at each one of these buffer systems. So let's first start with the protein buffers. This is the most abundant buffer system in the body, cells, and in the plasma. Remember, proteins consist of strings of amino acids, and the amino acids have an amino group, the NH2, and a carboxyl group, the COOH, over here. And we're going to use these to help in either accepting or donating those protons, those hydrogen ions, in order to keep pH constant. So the carboxyl group, in other words, acts like an acid when needed be, and it can release the hydrogen ions into solution and thereby lowering pH. So let's say your pH gets too high, the carboxyl group will let go of a hydrogen ion and thereby neutralizing that basic pH. The amino group can act like a base, removing hydrogen ions. That is, they can take up hydrogen ions, removing them. So if the blood gets too acidic, the amino acid group, or the amino group, excuse me, can kick in and remove those hydrogen ions. And we can see that here in this slide. So if your pH falls, for example, we've got too many hydrogen ions. So here's that amino group, takes on that hydrogen ion or binds with that hydrogen ion, thereby removing the hydrogen ion or accepting that hydrogen or proton, and therefore neutralizing it and absorbing that hydrogen ion. Whereas if the pH rises, then we've got too many hydrogen ions, too few hydrogen ions, and so that way we can take that hydrogen ion of the carboxyl group, put that into solution, thereby lowering the pH because we're adding hydrogens or donated hydrogens to the solution. Hemoglobin, we saw, works really well as an intracellular buffer. When we did carbon dioxide transport, we saw that carbon dioxide in water is made into carbonic acid, and those hydrogen ions that dissociate from the carbonic acid bind with the hemoglobin. Well, this is what neutralizes or keeps the pH from becoming too low by a buildup of hydrogen ions, is that binding with the hemoglobin. So the hemoglobin is a buffer. combining with the hydrogen ions, removing the hydrogen ions from solution, so therefore our pH doesn't drop too low. Another buffer system is the phosphate buffer system. This works primarily inside cells. It's all about either tipping this equation to the left or to the right, depending on which direction we need to move. And so it involves the dihydrogen phosphate or monohydrogen phosphate, which can act as either weak acids or weak bases to neutralize the bases or acids, as we can see. So think of it this way. It's the easiest way to look at it is, notice the double arrows. The equation can move in either direction. So let's say my pH drops and I have a huge increase in hydrogen ions. Think of that as shooting this side of it up. And so we've got this side of the equation tipped up, which means the equation is going to run to our left. And those monohydrogen phosphates, dihydrogen phosphates, even the trihydrogen phosphates are going to attach those hydrogen ions onto their molecules, thereby removing them from solution and neutralizing all that acid. Whereas if, let's say, hydrogen ion concentrations drop too low, so now our pH is high, since this side is really low, the equation is going to run to the right. And thereby I'm adding hydrogen ions to the solution. I'm donating protons, so to speak, so it's adding hydrogen ions, thereby lowering the pH. And so it's just a matter of which way this equation tips, right or left, to balance out the high or too low levels of hydrogen ions. The carbonic acid bicarbonate buffer system is the most important of the buffer systems that we'll be looking at because this is the one that's going to affect blood pH the most. It's a matter of looking at this equation that we've been seeing all semester, right, where we've got carbon dioxide and water forming carbonic acid, and that forming then bicarbonates and hydrogen ions. Now all we have to do then is tip this equation either left or right to adjust the hydrogen ion concentrations here. So imagine if we have too much acid, that is, the hydrogen ion concentrations shoot up. Well, now that shoots this side of the equation up, so it's going to run downhill, so the equation is going to run to the left, forming carbon dioxide and water. Which means then that I'm removing hydrogen ions if I get the equation run to the left. And I can exhale those CO2s to get rid of it. Now if I have too little acid, then the equation is going to run to the right. It's going to run from the carbon dioxide and water to finally make bicarbonates and hydrogen ions. And so if my pH drops, or my pH increases, that is, I have too low a hydrogen ion concentrations or too little of acid, I can make the equation run to the right, adding hydrogen ions and therefore lowering pH. Now there are problems with the chemical buffer systems in that they only provide a temporary solution to acid-base imbalances. They don't eliminate the hydrogen ions, they're just taking up the hydrogen ions. So if there's too many hydrogen ions, they just tuck them and add them to the molecule. So be it phosphates or bicarbonates. I add a hydrogen ion to bicarbonate, I'm just going to make carbonic acid and then CO2 and water. And so I'm just tucking that hydrogen away and removing it from the solution. But I haven't gotten rid of the hydrogen ions. So that's one problem. And the other is if all those molecules are busy tucking in those hydrogen ions and accepting them, again, since we haven't got rid of them, those buffers get used up. We run out of buffers then because they're all busy holding on to a hydrogen ion to make sure the pH doesn't get too low. So our supply of buffers is going to be limited, and so they're only going to work in the short term or small changes in blood pH. So when we get to bigger changes, that's when we need to turn to the physiological buffers, and that's where the respiratory and urinary systems come into play.